All right, good morning, everyone. It's good to see you. I'm Maggie Chen. You can just call me Maggie, and I'm going to be your instructor for the Understanding Our Environment Lab, ENVS 100 Lab for the spring semester. A little bit about me, I'm finishing up my second year in the MSCM program here at USF. This is my second year teaching these labs, but there's still a lot for me to learn. So if you do have any questions, I know that I've been told before that I tend to talk fast sometimes. So if I ever do, you can just type it into the chat. Could you repeat that? Or if you have any questions, you can just let me know and I'll get to them, or just unmute yourself and you can just ask me any of these questions. But okay, let me just share my screen and go over our campus page. So on our main Canvas page, right in the main home page, there is our lab schedule. It's tentative, but it's I think there's not going to be any more changes from here, but I will let you know if there is. So last week was our first week of classes where there was no lab. This week for February 1, the week of February 1, we're going over Lab 1, data gathering, analysis, and reporting. So we're going to be, I hope you all have Excel. If there's any problems downloading that, please let me know. But we're going to be making 2 Excel charts and also just looking at how to use different measuring tools. Next week for Lab 2, we're going over understanding experimental design using SymBio, a computer program to just look at the different variables and just within an experimental procedure and what could we do to change things around. But the week after that, there's no lab because of President's Day. So there's that 1 week break. Then the week of February 22, we're going over Lab 3, again using SymBio, that same computer program, and our experiment is on Isle Royale population ecology. So we're just looking at different populations, like the predator-prey relationship, how does that affect each other, and what does different resources and the amount of resources, how would that affect different populations. For the week of March 1, we're going over Lab 4, which really a nice transition into population growth and the ecological footprint, but looking at our own ecological footprint. And that's going to lead into, we're going to start looking at lab or part 5A for our water resources, at our own water resources, how much water do we use? And then that'll lead into Lab 5 for the week of March 8, where we're just going to do some calculations and then just looking at ourselves personally and comparing it to the class, how much water we use. And then there's spring break for the week of March 15th, so no lab. And on the week of March 22, we have Lab 6, which again, just leading right into water quality, looking at different water quality parameters like dissolved oxygen, temperature, turbidity. How does that affect the organisms that live in water? How would that affect us? And then after that, there's no lab because there's an Easter holiday. So really well spaced out this semester, I think, with like having labs and then no labs. But Lab 7 is in the week of April 5th. Again, looking at more water quality parameters like conductivity, salinity, desalination, going over a little bit about water treatment and what does that mean for the water that we use. For Lab 8, on the week of April 12th, we're now looking at moisture in our atmosphere and looking at weather data. How do we gather weather data? How would we analyze that? And then the week of April 19th, there's no lab. We say it's a semester check-in, but it's really if you have any labs that you didn't get the chance to turn in, that you have questions on that you didn't really know how to do, this is a time for you to turn those in, ask any questions, and just give you a little break right before the end of the semester. And then on the week of April 26th, for Lab 9, we're going over the greenhouse effect. So CO2 concentrations within our atmosphere, how do greenhouse gases change the temperature within our atmosphere and then change the environment that we live in. And so the semester check-in actually replaced Lab 10. So there is no Lab 10, but The very last lab we're going over is lab 11, where we're using Google Earth Pro to look at different energy resources and the amount of land that they take up, the pros and cons of each. So analyzing coal, wind, and solar energy. And then for May 10th, week of May 10th, and then there's no lab for that week. I think that's when you guys start going into your finals week. But that is basically the schedule for this semester. I think there's a lot of breaks in between that's really helps spaces everything out for us and gives us like a little break whenever it could get like, you know, a little bit much at times. So all of the information for the labs can be found if you scroll down at these links for each of the labs. So lab 1, if we're going to click into this. So even though I sent out my announcement with the background materials and where you can take your pre-lab quiz, in these links you can also find other background information like how to use, or just like a brief intro on like Excel, and then more of the like that extra data, extra supplies for this lab, and where you can turn in your lab 1. So there's more information on that in each of these links. So if we go back to the home page, if we scroll down a little bit more, actually, did anybody have any questions about the schedule or how anything like that? No? Okay. So if we scroll down a little bit more, this is where you can find our instructor online learning pages. And right in the middle, under mine, you can find additional information on how to contact me, things like that. So you can reach me at this email, mchen86@usfca.edu, or you can just message me right on Canvas. Or you can text me. This is my phone number. If you have any questions that you don't think that you really need to email me about, you can just text me. And so for the lab policies for the Wednesday AM and Thursday AM lab. So attendance of Zoom sessions is mandatory unless you have a time difference greater than 3 hours to US West Coast. So if this applies to you, if you're suddenly moving in the middle of the semester or anything like that, and you find yourself in a different time zone, you can just email me, we can work something out, and then you can take this lab asynchronously. So this, every single lab is recorded, and the recording is going to be uploaded by the same day that we have lab. So this recording is going to be available probably a couple hours after we end lab, and you can watch those asynchronously, or if you're doing lab asynchronously, or if you have any questions about anything that we did throughout lab, you can just refer to the recording. Entering a lab session 5 minutes late will result in a tardy attendance mark. Entering the lab session 15 minutes late will result in an unexcused absent. Have your camera on and microphone muted. If you have any questions, just unmute yourself or type it into the chat. Attend lab in an appropriate setting, wear appropriate attire. Pre-lab quizzes are due at the start of lab. So almost every single lab, there's going to be a pre-lab quiz, and that's due right as we start. If we don't have a pre-lab quiz for that lab, I'll let you guys know and just send out an announcement to say that we don't. Assignments will be available at the start of lab. So assignment for lab 1 is already open, and they will be due the next following lab session. So week 1, lab 1 assignment is due by the start of lab 2 next week. So regrades of assignments may be requested. However, the entire assignment will be subject to regrading. So if you think that maybe I misunderstood your submission, or that I didn't really get what you're trying to say, I can look over your assignment, but then I could also just look, and I would regrade everything as well. So that's that. This is the link to where you can find the Zoom recordings. I try to keep captions on too. I know that I tend to get like a lot of students from SDS, so captions really help. And then more additional resources at the bottom. If you didn't get Microsoft Office, here's the directions to do that. Google Earth Pro download, that's going to be later on in the semester for one of our later labs, but our Zoom meeting link and more of that type of information is down here. We're going to be using this exact same link every single lab. So once you have this one down, you don't really, nothing's going to change with that. But if you do need to find it, it's going to be on this page. And that is basically it for my online learning page. Did anybody have any questions about the lab policies or about anything like that? Okay, so now we will get started with our lab 1. Okay. Okay. So, lab 1, gathering, analyzing, and understanding scientific data. It's important to have context for what we're looking at. So taking into consideration units, reporting the important amount of figures in our final answer. That's what we're going over today. And this PowerPoint was made by our lab manager, Darren Chen. So today we're going over 3 modules. First, unit conversions. We're making sure that our final answers and the units that we're working with are all consistent throughout the entire problem that or the entire situation that we're looking at. Then we're looking at significant figures, which numbers are important to us, what do we need to know from this situation, and scientific notation. When we have numbers that are too big or too small, how can we report those numbers? And finally, we're going over accuracy and precision. What's the difference between those, and what do they mean in terms of us looking at different examples? So first, unit conversions. Units describe the physical quantities that we're interested in understanding in environmental science and management. So there's different types of units. Some basic units are length, volume, mass, and time. So length, we have as an example, meter, which can be abbreviated as lowercase m. Volume could be, an example of that would be liter in uppercase L. Mass could be grams in lowercase g. Time, as an example, would be second or s. So we can also have mixed units like velocity, flow, and concentration. So as an example, for velocity, we can have meters per second, and we would write that as lowercase m \s. So these are some of the different units that we could see. But now if we were going over prefixes, more like there are also different types of prefixes that we can indicate in our final answer. And we know that some of them are mega, kilo, deci, centi, milli, micro, and nano. So if we're looking at centi as a prefix, it could be abbreviated as lowercase c. The multiplier of that is 0.01. And what we can rewrite it as in the power of 10 is 10 to the negative 2. So how would we get this power of 10? This negative 2 indicates, or this power of that exponent indicates how many spaces past the decimal point our number is. So this 2 is this negative 2 is the 2 spaces to the right of our number. So this negative indicates that it's a small number. But now if we're looking at mega as a prefix, it can be abbreviated as uppercase m, and it's a multiplier of 1 million. The power of 10 is 10 to the 6, and this 6 is how many spaces, again, past the decimal point it is. So it's 6 spaces to the left, and that means that this is a really big number. So again, at the bottom here, if mega is 10 to the 6, then 1.2 megatons can also be written as 1.2 times 10 to the 6 tons. So they mean the same things, but you can just write it in different ways. Dimensional analysis. With dimensional analysis, we can multiply the quantity by conversion factor. So if we wanted to change our whatever figure, whatever number that we're given in a certain unit to a different unit, this is what we can do. So say we're given 0.08 centimeters and we wanted to change it to meters. So we can write out first 0.08 centimeters, and our conversion factor, we know that in 1 meter there's 100 centimeters, and we can multiply that by 1 meter over 100 centimeters. The centimeters would cancel out, and they would leave us with the units of meters. So we would just multiply across 0.08 times 1 divided by 100 is going to equal 0.0008 meters. These slides are available to you on the Lab 1 link. So if you wanted to refer to this later, then that's where you can find those slides or these slides. So unit conversion. Numbers are often reported with inconsistent units. So what we can do from here when we do calculations is that we would want to convert them into the same units so we can have our sum, our final answer to be in the same unit. So as an example, the length of the canal is 2.31 kilometers and is connected to a stream that is 150 meters long. So already we're looking at 2 different water bodies and they're in different units. One is in kilometers, one is in meters. And they're asking us what's the total length of this water body. So what we can do from here is do our unit conversions and convert to meters. So we're going to convert that 2.31 kilometers to meters. And we can do that by first writing out 2.31 kilometers. Our unit conversion, 1 kilometer will have 1,000 meters, and we would just multiply that across. So 2.31 kilometers, the kilometers here would cancel out with the kilometers at the bottom of this fraction. So we would multiply 2.31 times 1,000 divided by 1 and get our final length of our canal in meters as 2,310. And now that we have this number, the length of our canal in meters, we can just add it to our stream where the units are already in meters. So we have our 150 meters is equal to 150 meters. And if we add that together, the total length of our water body is 2,460 meters. So this is one simple way to do unit conversion to get our final total length in the same units. Now we're going over significant figures in scientific notation, but did anybody have any questions about unit conversions or anything like that? Okay. So significant figures, what are significant figures? They are whole numbers from 0 to 9. They are also the numbers that are important to us when we're looking at a certain context. And there's a lot of different rules. And again, these slides are available to you on Canvas. So if you want to refer to them later, you can. So these are a lot of rules. So first, we're going over how non-zero figures are always significant. So as an example, 9,683, they're all non-zero figures. And that means that they're all significant. So there is 4 sig figs in this number. But for zeros, there are different rules for which ones are significant or not. So leading zeros that come before all non-zero figures and are not significant. So if we have 0.0005, these zeros before the 5 aren't significant. So we would just count this 5 as the sig fig as our 1 sig fig. Captive zeros that are between non-zero figures are always significant. So 15.40002, these captive zeros we're going to count as a part of our sig figs because it's between the 4 and the 2. So therefore, there's 7 sig figs in this number. Trailing zeros that come after all non-zero figures are only significant if a decimal point is present. So if we have this decimal point within our figure, that means that the zeros that come after are significant. So this number would have 6 sig figs. But then trailing zeros without decimal points can sometimes be significant. So it's a little bit vague when we have a number like 50,600 kilometers, which could be 3, 4, or 5 sig figs because there's no decimal points. So we can just be more clear about how many sig figs that we have if we use scientific notation. And I'll be going over how to do that in a little bit. But another rule for sig figs is that exact numbers via counting or definitions are infinitely significant. Don't worry about their sig figs. So that's like when we did our unit conversions where 100 centimeters are in 1 meter. So those you don't have to worry about their sig figs. So scientific notation. Scientific notation is a way to report numbers that are really big or really small. And scientific notation, we can use this to indicate how many numbers are significant in our final answer. So it's just another way to write numbers. And as an example, 0.00001 can be rewritten as 1 times 10 to the negative 5. They mean the exact same thing and they have the same significant digits. And so this negative 5 again is how many spaces past the decimal point that we're looking at. So if we wanted to rewrite 200,000, how many, or if we're asking how many sig figs is intended by this number, we're not really sure. So it depends on how you would write it in scientific notation. If we write it as 2 times 10 to the 5, it tells you that there's only this one sig fig in our number. If we write it as 2.00000 times 10 to the 5, this number tells us that there's 6 sig figs. And if we write it as 2.00 times 10 to the 5, then this tells you that there's only 3 sig figs. So it really depends on how you write your number to indicate which, like how many sig figs there are and which numbers are important to us. So we can write really big numbers or really small numbers using sig figs or scientific notation. So the sun has a mass of 1.988 times 10 to the 30 kilograms. And it's much easier to write this than writing this big number with all of the zeros at the end. So scientific notation can help us out when we're doing calculations and using really big or really small numbers. So, but the breakdown of scientific notation of a number would be, as an example, we have the mass of the sun again. So our 1.988 times 10 to the 30 kilograms. So in the front here, these are our significant figures. It's going to tell us how detailed our measurement is. And that's the first part. The 10 to the power of 10 to the 30, this exponent gives us an idea of scale. So 10 to the 30, we know that's all of those zeros past the decimal point. But if it's 10 to the negative 30, we would know that that's a really small number. And at the end, we have our units, which tells us what we're measuring. And units are very important because if we don't have them, we don't really know what we're looking at. Is it in kilograms, which is very different from grams? Is it in miles? So whenever you're reporting your final number, make sure to put in the units too. So, all right, so it's your turn. So the first part of a number in scientific notation consists of the significant digits only. If we wanted to rewrite 0.00045 in scientific notation, how could we do that? I'll give you a couple seconds to do that. You can either unmute yourself or type it into the chat, but how would we do this? We'll give you a couple seconds. Perfect. So from Sebastian, 4.5 times 10 to the negative 4. That's exactly how we would rewrite this, because we would want to count the spaces past the decimal point, and we know that these two numbers, 4 and 5, are our significant figures. So we would write it as 4.5 times 10 to the negative 4. But now we wanted to rewrite 4,500 in scientific notation. How could we rewrite this? Perfect. Thank you, Sebastian. Okay. Oh, I accidentally stopped sharing. Okay. That did not mean to do that, but yes. If we wanted to rewrite this in scientific notation, it would be 4.5 times 10 to the 3. So these are our 3 spaces past the decimal point. But great. Okay. So some other ways to write scientific notation. So in some calculators, like in your scientific calculator or in some computers, they report scientific notation with the letter E. So yeah, as an example, you don't need your scientific calculator today, but other calculators online like Desmos, they would have E, or that would just mean the 10 to the 5 or the power, 10 to the power of whatever. But if we were looking at 700,000, we can rewrite this in scientific notation as 7 times 10 to the 5. They mean the same thing, and it also means the same thing as 7e plus 5. And same thing for really small numbers like 0.00007. We'll rewrite this as 7 times 10 to the negative 5, or 7e minus 5. They all mean the same thing. The e just indicates the 10 to the whatever. So sig figs and calculations, there's specific rules to sig figs when we do our calculations. So with the calculations that we're using data with, there's a specific level of uncertainty. So you have to report your final answer with the appropriate level of uncertainty. As you're doing your calculations, there probably is going to be like more decimal points or more decimal places that's been given to you as you do it. So at the end, you're just going to have to round to the right amount of sig figs. And sig figs are only important when reporting your final answer, but you should use all available digits, both significant and insignificant, during intermediate calculations. So that's what you're going to do. At the end of your calculations, that's when you look at your initial numbers that you were working with, looking at those sig figs, and then rounding your final answers to have the right amount of sig figs. So sig figs in calculations, if you're multiplying or dividing, is you're going to report sig figs equal to the quantity with the smallest amount of sig figs being multiplied or divided. So as an example, if we have 1.23, there's our 3 sig figs, and this number 4,567.89, there's our 6 sig figs. We're going to go with the smallest amount of sig figs that's being multiplied or divided. So we're going to go with our 3 sig figs. So our calculator would give us 8 sig figs when we multiply them together. And we would want to round this to our initial lowest amount of sig figs, to our 3 sig figs. So we would rewrite that as 5,620. But to make that even clearer, we can also use scientific notation and rewrite this as 5.62 times 10 to the 3. So scientific notation would clarify sig figs for us and also just make it easier to know, like easier to use later on too. But for addition and subtraction, there are different rules for this too. So we would want to report the decimal places equal to the least number of decimal places in the values being added or subtracted. So the least number of decimal places in the original numbers that we're looking at. So if we're trying to add 1.234 plus 567.89, in this original number, this first number, we have a number with 3 decimal places. In our second number, we have our 2 decimal places. So we're going to report our final number to the least number of decimal places. So that would be our 2 decimal places in this number here. So if we add these two together, our calculator would give us 3 decimal places. But in our final answer that we would report, we would just give these 2 decimal places at the end. So the least number of decimal places that's being added or subtracted. Did anybody have any questions about sig figs when we're doing calculations? Okay. So now, going over accuracy and precision. What are they? So accuracy is exactness or how true something is. This is generated by calibration to a known standard or value. What this would mean, as an example, if we're comparing a speedometer in your car versus a police radar gun. So when you buy your car, your speedometer is probably calibrated once. You don't go in for yearly checkups and calibrate your speedometer. You probably won't ever have to do that. It's just like once you drive it off of the lot, then your speedometer is probably never calibrated again. But a police radar gun has to do annual calibration checks so that it holds up in court. So police radar guns, they would they have regular calibration and they need to have like a certain standard that's calibrated to. So if we're comparing the speedometer in your car versus a police radar gun, the more accurate of the two, how true it is how calibrated it is. So it would be the police radar gun, which is calibrated more often. But now if we're looking at precision, it's the quality of being well-defined, the level of detail presented. So the more detail that our instrument gives us, the more precise it is. So this is generated by higher quality instruments and sensors. So more sig figs equals more precise instrument or measurement. And if we're going to compare 2 different measurements, we could look at a bathroom scale versus a laboratory analytical balance. So for your bathroom scale, it probably maybe gives us up to 1 number past the decimal point, maybe at best. But if you say I weigh myself and I eat an Oreo and then I weigh myself again, it probably won't register that on my bathroom scale. But on the laboratory analytical balance, it gives us up to 2 decimal places and it's very sensitive and it gives us more detail, more sig figs. So comparing the 2 between a bathroom scale versus a laboratory analytical balance, the more precise measurement would be given by our analytical balance. So the more detail that's given to us, the more precise that instrument is. So now if we're looking at different figures, we can say that this top left one is accurate, but it's not precise. So precise is the level of detail given to us. And if we're looking at this figure on the upper right, it's precise, but it's not really accurate. Accurate would be how close to the center we would want, so how true it is. In the lower left, we can see that it's neither accurate nor precise. And the bottom right, we can see it's both accurate and precise. Data precision. So if we are measuring the length of fish to study the effects of water pollution on fish growth, how are these measurements different? So if precision is how much detail we're given, then looking at these 3 different figures, 3 centimeters, 3.0 centimeters, and 3.00 centimeters, the more detail that's given to us, the more precise that measurement is. So that would mean more sig figs, more precise measurements. So this would be our most precise, and this would be our least precise. So now if we're reporting measurements, and if we're going to be measuring a quantity, the rule is report the digits given by your measurement tool. So now if we have that same fish and we are measuring it using two different rulers, which one is more precise? So say we're looking at this top ruler in centimeters. We can see that it looks like it's not quite 4 centimeters, but it's probably past 3.5. So maybe we could guess and say it's 3.7 centimeters, but we're not really sure because that amount of detail isn't given to us. But now if we're looking at this bottom ruler where our millimeters are also given along with centimeters, we can say more precisely that it's actually around 3.8 centimeters. So that level of detail is given to us. And now we can say, comparing the two, the ruler that gives us more detail would be the more precise ruler, more precise measurement. So this ruler at the bottom would be more precise. And that is an example of how to look at precision between different instruments. Did anybody have any questions about accuracy or precision or anything like that? Cool, cool. Okay, let me just bring us back to the Canvas page, and we'll go over how to use rulers and calipers when we're measuring different leaf samples. So let me just share my screen again. Okay, so if we're going back under pages, at the bottom for lab 1, this is a short, around 9-10 minute video about how to use a ruler and a caliper to measure leaf samples, the length and width. You don't need a caliper to do this. Later on, we're just going to be making Excel graphs using the data that our lab manager has provided for us. So let me just play this video for you all.
Raw data of leaf sizes using two different measuring tools, a ruler and a digital caliper. When the term raw data is used, it simply means data taken from a direct measurement or data that has not yet been processed or run through any calculations. You will be using this raw data to calculate more advanced values and create graphs later on in this lab exercise. Here is our standard rule.
Can you all see this video or?
Yes.
Oh, okay, awesome.
Yeah.
Okay, awesome. Okay.
Ruler. On one side, it measures English units in inches. And on the other side, it measures metric units in increments of centimeters. So each one of these numbered lines is your centimeter. We also see that our metric side has these smaller marks, and these are in increments of millimeters. So one of these small marks is a millimeter. And as we know, 1 centimeter consists of 10 millimeters. Therefore, we would say that this ruler is able to determine the length of an object to the nearest millimeter. Here is our digital caliper. It has jaws that can be opened and closed to measure the length of an object. It has a digital display that is controlled by an on-off button. And it can display its measurements in either inches or millimeters. And those can be changed with this top button. There is a third button that is labeled 0. This button is used to calibrate the caliper. The caliper is capable of giving us very accurate data, but first we need to be able to trust that it understands us. For instance, we can all agree on what the measurement of 0 millimeters looks like. that should mean that there is absolutely nothing between the caliper jaws and that they should be completely closed shut. So while we have that, the caliper jaws being completely closed, what do you know? Our display does not read 0. So that means the caliper does not understand what we mean by 0. It has its own idea of what 0 means at this time. So we can fix that by making sure, again, our caliper jaws are completely closed, and then pressing the 0 button, telling it that this is what 0 looks like. So that means what we have done is that we have calibrated the caliper to a known standard value, that value being 0. Now, these calipers are on the same page as us, and they are ready to go ahead and get measuring. Okay, so these are the 3 species of plant leaves that we will be measuring for our lab today. And right here, this is blue blossom, this is California bay laurel, and this right here is arroyo willow. And all 3 of these are native to California, to Northern California, and they can be found actually all over campus at USF here. So what I will be doing today is just demonstrating in this video measurement techniques for our ruler and our caliper with the California bay laurel. So in our procedure, what it outlines for us to do is to take the length and width measurements of the California bay laurel 3 times, and each one of those will be independently sampled, meaning that we're going to place our measurement tool at, say, the widest spot to get the width, and that looks like it is about 30 millimeters, and then we take it away. We remove the measuring tool and then place it back on the leaf and take another reading. And this looks like it is about just a little beyond the 3 mark there, so I'd say right there 31 millimeters. And differences like this can arise just from the positioning of the ruler or the measurement tool on the leaf. It can arise from just how we are looking at it, how we are viewing the ruler. Right now, I would ideally like to be my head right above the ruler, but I have the camera in the way, so it's a little difficult for me to get the best reading possible. Differences like this can occur just between different people measuring the leaf. If you're working with a lab partner and you're measuring the same leaf 10 times over each or something like that, you're definitely bound to get some differences. I would say some differences between your measurements and your partner's measurements. Now our final reading, our 3rd reading on the width of this leaf is, it might be a little difficult to see on the camera, but it's just between the 3 centimeter mark, or the 30 millimeter mark, and the 31 millimeter mark right after it. But to my eye, I would just say that it looks a little bit closer to the 30 millimeter mark, so we will call it that. So our readings will say 30, 31, and then 30 again millimeters. Okay, now let's repeat the same process with our calipers. So we're going to once again be looking at the California bay laurel, and we are going to be using our calipers to measure the width and the length. So We've got them all zeroed out. They've been calibrated, and they're ready to roll. So let's start, and our first measurement of width will read just around 31.1 millimeters. Take the calipers away. I like to close them to make sure that I'm not cheating and using the same measurement again. And reopen them. And let's try to find that widest point on the leaf. Okay. Here I'm measuring at 30.6 millimeters. Remove the calipers to maintain independent measurements. And final reading, 31.2 millimeters. Here I've gone ahead and drawn a rough rectangle shape using the measurements of the leaf and using the length and width measurements of the leaf. And this is how we're going to calculate leaf area. So with that in mind, take a look at the shaded in red space around the leaf, and you will use this visual aid to assist you with some of your post-lab questions. In contrast, here is an oval shape that I've drawn around the leaf using its length and width measurements. It's not a perfect oval, but as you can see, there is much less red shaded area between the oval and the actual shape of the leaf itself. So once again, use this in conjunction with the drawn rectangle and to answer some of your post-lab questions. So I'll leave you with that, and rest assured that the same process has been carried out with our blue blossom leaf as well as our arroyo willow leaf.
Okay, let me see. Okay. If you have Wi-Fi issues throughout lab and if you have to just suddenly join, that's totally fine. These recordings are available after, like a couple hours after lab, so you can just watch whatever parts that you've missed. So it's totally okay. Okay, so now we've looked at how we can use either a ruler or a caliper to measure different leaf samples and get their lengths and widths. And now we're going to be working with the data, these measurements, to make some Excel graphs. So right underneath this video, there's a link to get your Microsoft Excel sheet with these measurements, and we're just going to click and open that, and we'll be using with this data. So I'll just be giving you like a couple of seconds to do that. But when you do open it, it's going to look like this. So on the left side of this first page, we can see that we have our ruler measurements between the 3 different leaf species. So blue blossom, Caliph Bay laurel, and arroyo willow. It's been measured 3 times for each leaf for their length and width. And these measurements are available to us here. Our caliper measurements is on the right. And again, the same 3 leaf species measured 3 times for both their length and width. And these are their measurements here. Does everybody have the Excel sheet open by now? Okay, awesome. So what we're gonna wanna do is get our leaf area and do some calculations on Excel. And from here, if we wanted to get the leaf area for a rectangle, we know that for that, we would just multiply length times width. And one way to get this leaf area is we can start our equation by first typing in an equal sign. And if you can't really see this, what I'm typing into the cell here, there's also this formula bar in white up here that you can refer to, where the equation is just going to pop up. But if we're going to want to get our leaf area, we can just start our equation with an equal sign and then select our length, 44, shift 8 to get our asterisk or our multiplication sign, and then select 25, our width. And then we can hit enter. Another way for us to get this leaf area, we can also use different functions in Excel. So one of them, if we again start our equation with the equal sign and just type in product, or even when you start typing in, there's going to be a drop-down menu with different functions that you can use. So product would be one of them, one of the first ones, and you can select that. And it would give us this function and it would start it for us. Our first number that we would want to multiply by would be 43. We'll just put in a comma and then select our width at 24. And then we can just hit enter. So these are two different ways for us to get the product. Either one works. But from here, since you won't really need to type it in every single time, if you can just hover over this cell that we've just typed in, and then this plus sign where our cursor is, when it changed from white to black, when it's a black plus sign, you can just click and drag, and it would just autofill those formulas for us so we don't have to type it in every single time. And this is the leaf area for our rectangle. But now if we wanted to find our oval leaf area, the equation for that is pi over 4 times length times width. So we would just type that in. So we'll again start our equation with our equal sign. For the pi over 4 function, we'll just put that together in parentheses. So started with our parentheses and capital PI. Our function would pop up in this drop-down menu, and we can just select that. It would start our parentheses, but we're just going to end that parentheses. So capital P, capital I, and then we're just going to end that parentheses and divides or backslash by 4. And then we're going to just finish that first part of our equation by putting in one final parentheses at the end. And then we're just gonna shift 8 to get our asterisk, our multiplied by sign, select our length, and then our asterisk again, and to multiply that by our width. So this is our final equation, pi over 4 times length times width. Now we can just hit enter, and it would give us our leaf area. And again, if we wanted to just autofill the other cells at the bottom, we can hover over this main cell. And when our black plus sign in the corner here, when it shows up, we can just click and drag, and it would just auto-populate these other cells at the bottom with the numbers that we want. Did anybody want me to go over that again, or did anybody miss those equations? Okay. Awesome. So from here, we can see that these numbers are really big, but if we wanted to just format them, we can do that using scientific notation. So we can first do, we're just gonna select the numbers that we wanna format. So I'm gonna select these cells here, and at the top of this Excel sheet, under the Home tab in the middle of this section, we can see that there's this number format. If you click on the little arrow, a drop-down menu is going to come up. And all the way at the bottom, right underneath fraction is scientific, and it's going to format the numbers into scientific notation for us. So if we just select this, it should change our numbers into something more manageable. So from here, there's a chat. Can you repeat, please? Yes, of course. So for the rectangle leaf area, I can just redo it again. So first we're gonna start our equation with the equal sign, and we'll just select the length that we want, put our shift 8, our asterisk, multiply by our width, and we'll just hit enter. And that'll give us our leaf area for a rectangle. We can then hover over this cell in the bottom right corner where that little dot is. And when our plus sign that looks like a little black plus sign, we can just click and drag, and it would just autofill these other cells with the same formula so we don't have to type it in over and over again. And the equation for oval leaf area, will again start it with an equal sign. For the first part of our equation, we'll just put that in parentheses and capital Pi for our pi function. And when we click on that, it'll start our parentheses, but we're just gonna end that parentheses, our first pi with our 2 parentheses. We're gonna divide that, or backslash, divide that by 4, and then end that entire first part with another parentheses. Then we're gonna multiply this by our length and width. So again, Shift 8 to get our asterisk. We're gonna select this length, our length for our sample, and then again, Shift 8 to multiply again using our asterisk and select our width. So from here, we'll just click enter. And then again, we can just click and drag so that these numbers just auto-populate, or these cells have just fill in with the formula that we just made. And then we can format this by selecting the cells that we want to change into scientific notation. Going back under the Home tab at the top here, right in the middle where it says number format, you can click on this drop-down menu, and all the way at the bottom, right underneath fraction, we can change it into scientific notation. Great. So one nice thing about Excel, is that if we don't want to retype things, we can just select the cells that we want, that we have the formulas with, Command C to copy, and Command V to paste. And they would just continue and auto-populate with those formulas that we want. So we'll just Command V, Command V. And so now all of these other cells are filled in with the formulas that we want, and it does the calculation for us. Another thing that we want to make sure is that we have these cells in the sig figs that we want. So some of them gives us this extra decimal place and gives us 3 sig figs instead of 2. So what we can do from here is just select these cells, and right underneath that number format, you can add or take out a digit. So you can either add or take out, keep it at our 2 sig figs. There's another question in the chat. Can you show how to auto do them all again? Sure. Let me just take out some of these. The auto populate thing, or just putting in the formulas, it's really just copying the cells with the formulas in them. So Command C, and then going to the cells that don't have any formulas in them, and Command V, or paste them in. And you can just Command V and just continue pasting in these formulas. Yeah. Did that help? Did anybody else have any questions before I move on? Okay, yeah, no problem. Okay. So again, we can just adjust the data that we're given to put it in the sig figs that we want. So if we look off to the side in our caliper measurements, we can see that some of them aren't in 3 sig figs. So again, we can just select those cells, add in or take out an extra digit to keep it at our 3 sig figs. We'll just add in, make sure it's our 3 sig figs. Same for here. So those 2 buttons are right underneath the number format. And now that we have it in our 3 sig figs, we can move on and calculate our averages. So we'll just start with our ruler measurements here. in the yellow or in the blue cell right underneath all of our data, we can start our equation with the equal sign. And we'll just type in AVE for like the first part of our average function. This drop-down menu is going to pop up and we'll just click on this average function and then select these 3 cells at the top. Once you've selected the cells that you want to get your averages from, you can just hit enter. And that should give us the average that we want. And again, from here, like how we did for the green cells, we can just hover over the corner of this blue cell until our black plus sign pops up, and we'll just click and drag, and it would do these calculations for us. And then we can also put these 2 numbers in scientific notation again. We can just select these 2 numbers, going back under the number format bar, and then just select scientific. And like how we did for the green cells, we can select the blue cells, the averages that we just made, and then Command C or copy. We can just paste. And that would fill in the other cells that we would want and get the averages for those. Again, we can also put these numbers back into or keep the 3 sig figs that we want. So we'll just select these cells, add in that 3rd sig fig. So we'll just select those, go back under this number format bar and just add in that 3rd sig fig right at the top here. And that is the main first section. Did anybody have any questions, or did you all want me to go over anything else?
I have a question about the 3rd table on the left.
Oh, this one?
Oh, no, sorry. Over to the left, the bottom for the Arroyo Willow.
Yeah.
There's the one yellow cell that says 20.0, so it's 3 sig figs, but why did we only use 2 for that table?
Oh, yeah, we can just take out that 3rd digit and
Okay.
keep it at 2 sig figs.
Gotcha. Thank you.
Yeah, thanks. Thanks for catching that. But yeah. Okay, did anybody else have any other questions? Okay, so now that we're done with this first part, we can just scroll over to the right and our summary table is given to us. So these are just the numbers that we calculated off to the left. And what we can do here is again, change them into the sig figs that we want. So for the ruler measurements, we'll just want to put that back into 2 sig figs. So again, right underneath that number format bar, we'll just take out all of these decimal points. Keep it at our 2 sig figs. Our average rectangular and oval area, we'll just select these cells, put them back into scientific notation. And then same for here. For the caliper measurements, it looks like they're already in 3 sig figs, so we don't have to do anything for that. And on the side for the average rectangular and oval area, for our calipers, we'll again just want to put them back into scientific notation. Great. So from here, with everything that we have in their summary table, we're just going to be making 2 column charts. So the way we can do that is first, we're comparing the ruler and caliper measurements, the average rectangular and oval area versus by each leaf species. So we're going to want to select these cells on the end, our leaf species, and to select cells that aren't right next to each other in the column, we can either, it depends on your keyboard, it's either Shift or Command. For my keyboard, it's Command. After you've selected it, I'm gonna hold down the Command key and select the average rectangular and average oval area and the data here, and then I'm gonna release my Command key. So once the data has been selected, I'm gonna go up to the Insert tab. It's right next to the Home tab in the top left corner. And when you select into the Insert tab, around the middle of this toolbar, we're going to have different charts, like recommended charts, but we're going to want to select into our column charts. And different column charts are given to us: 2D column, 3D column, 2D bar, 3D bar. So it's this very first 2D column, this clustered column chart that we're going to want. So we'll just select this. And our chart's going to be made. So let's just make this a little bit bigger. And now that we have this, we can see that we have our 3 different leaf species, but a couple of things are missing. So if we want to, for whoever is just looking at our chart for the first time, we would just want to make it clearer for them. So we would want to add in X and Y axes titles, make sure that the units that we were using are in here. So, and then change like our chart title to something more descriptive. So once our chart is selected, the chart design tab is going to pop up on the top, and all the way on the left, there's add chart element. If you select this, okay, there's a chat. Do we turn this in today, or can we complete it and turn it in before next class? So all of the lab assignments are due the following week, right before we meet for lab. So this, the Excel sheet, and a couple of questions that you're going to answer at the end is due by next week, like 15 minutes before we meet for lab. But yeah. So back to the charts. If we wanted to add in our axes titles, our X and Y axes titles, under add chart element, there's this drop-down bar. And if you hover over axes titles, we're given our primary horizontal, or our X-axis, or primary vertical. We're going to want to add in first the primary horizontal. When that pops up on our chart, we can just change it to something that's, what are we looking at? So we're going to change this to leaf species. And that is going to be our X-axis. But now we also want to add in our vertical axes title. So we're going to go back under add chart element and hover over axes titles. And what we just selected before was our primary horizontal, but now we're going to want to select our primary vertical, or our Y-axis. So we'll just select this option. And when it pops up on our chart, we can just change it to a different name. So we'll just select and say this is leaf area. And we're going to keep the units that we were using to make sure that it's on the chart somewhere, so whoever's looking at it knows what we were measuring or what units we were measuring with. So in millimeters squared, our units are in here. And now we can also change the chart title to something more descriptive. So like different leaf measurements using a ruler. Something like that. It doesn't have to be exactly like what I typed out, but it should be descriptive. It should tell whoever's looking at your title, what are they looking at? And this is the first chart. And the process to make the second chart for our calipers is the exact same thing. So now if we're going to just make the exact same column chart, we would again select our species, leaf species, blue blossom caliphate, and arroyo willow. I'm going to hold down my command key and select the data on the side here for our average rectangular and average oval area for our calipers. And I'm going to release my command key. So now that my data is selected, I'm going to go back into the Insert tab right next to the Home tab. Around the middle, again, going back to our column chart, our column key, we're going to click into that, and that very first clustered column option, I'm just going to click into that. And now my caliper chart, my caliper measurements are going to show up in this chart. Let me just make that a little bit bigger. And the same thing as we did for our ruler column chart, we're just going to add in our primary horizontal and primary vertical axes titles. So once our chart is selected, chart design, that tab's going to pop up. And again, all the way at the end, on the left side, where add chart element is one of the options, we'll click into that, hover over axes titles, and then select primary horizontal for our x-axis. And again, this is leaf species. Now we're going to want to add in our vertical axis title. So back under add chart element at the top, here we're going to hover over axis titles, and right underneath horizontal, primary horizontal, we'll just add in our primary vertical or y-axis. And we can change that name to leaf area, again in millimeters squared. And you would just change this chart title to something that indicates that it's our caliper measurements. But that is basically it for our Excel portion. These are the two charts that you're going to turn in. Now, let me just, did anybody have any questions or anything about the Excel portion? Okay, so let me just go back to our Canvas page. So in the same Lab 1 data gathering, analysis, and reporting page, if you go all the way to the bottom, you can upload your Microsoft Excel file and complete the questions for this lab. So if we click into this link, it's going to take us to where you're going to turn this in. You can, these are the 3 questions that you're going to be answering. It sounds like there's like some people have had problems or little issues like uploading answering these questions in a Word doc and then submitting it. If that's the same for you, you can either type it in to your Excel sheet or say, let me put this into student view. Say you wanted to submit your assignment. You would choose your file, your Excel file. You can add another file if Word doc and doesn't work for you. You can just answer the questions as a comment or write into your Excel sheet. But you're just answering these 3 questions about precision, accuracy. You can refer to the slides for the specific definitions, but that is basically what's due by the start of next week. And that's basically it for Lab 1. If anybody had any questions, you can stay after class and ask them to me. But thank you all for coming in. I know it's probably been a pretty stressful first 2 weeks, just starting school online, but hope you have a good rest of your day. Yeah, thank you. Thank you.
I had a question.
Thank you. Sure, yeah.
What was the title for the what is it? The vertical axis on the first chart? Is it the same as the second one?
It's a leaf area in millimeters squared.
Okay, so it's the same. Perfect.
Yeah.
Thank you.
Yeah, thank you.
I have also one question. We're
Sure.
going to use the same Excel sheet that provide on Canvas, right? To do graphs and all, right?
I'm sorry, can you say the second part of your question?
To doing graphs and all